In our first session, we looked at bioinformatics as a field, what it is, why it exists, and the databases we rely on. Today, we go one level deeper and start with the biology itself. This session is part 1 of Molecular Biology Fundamentals, focused on DNA, the genome, and genes. We'll look at how cells are organized and where DNA physically lives, the chemical structure of DNA itself, how the genome and genes are organized within it, how DNA copies itself every time a cell divides, and what happens when errors, mutations creep into that process. By the end, you'll also write your first real lines of Python code, working directly with DNA sequences. This is the biological foundation everything else in the course builds on, so let's take our time with it. Here's what we're aiming for today. First, understanding how cells are organized, and specifically the difference between prokaryotic and eukaryotic cells. Second, understanding the chemical structure of DNA, what it's made of, and how it holds together. Third, understanding how the genome is organized into chromosomes, genes, and the regions in between. Fourth, understanding how DNA replicates itself, the process that has to happen accurately every single time a cell divides. And fifth, understanding what mutations are and why they matter biologically. This is a lot of foundational vocabulary, but each concept builds directly on the last, so don't worry about memorizing everything at once. We'll reinforce it with Python exercises at the end. Let's start with the basic unit of life, the cell. Every living organism is made of one or more cells, and cells come in 2 broad categories. Prokaryotic cells, found in bacteria, are simple and small, with no internal compartments. Their DNA floats freely in the cell, in a region called the nucleoid. Eukaryotic cells, found in animals, plants, and fungi, including us, are larger and more complex, organized into specialized compartments called organelles, each with a distinct job. Think of a prokaryotic cell like a single open-plan studio apartment, and a eukaryotic cell like a house with separate rooms for separate purposes. A kitchen, a bedroom, an office. The most important room for us in this course is the nucleus, which exists only in eukaryotic cells and is where the DNA is safely stored. Since this course focuses heavily on human biology, we'll be spending most of our time thinking about eukaryotic cells. Let's zoom into the eukaryotic cell a bit more since that's what we'll focus on. Inside the cell membrane, you'll find several organelles, each doing a specific job. The mitochondria generate energy, the endoplasmic reticulum builds proteins and other molecules, and so on. But the organelle that matters most for this course is the nucleus, a membrane-bound compartment near the center of the cell. This is where almost all of the cell's DNA is stored, tightly packaged and protected. Think of the nucleus as a vault, and the DNA inside it as the master blueprint for the entire organism. Every instruction needed to build and run the organism is written there. A small amount of DNA also exists outside the nucleus, in the mitochondria, but the vast majority, including everything we'll study in this course, lives in the nucleus. Whenever we talk about the genome going forward, we mean this DNA safely housed in the nucleus. Now let's get into the actual chemistry of DNA. DNA is built from repeating units called nucleotides. Each nucleotide has 3 parts: a sugar molecule, a phosphate group, and one of 4 nitrogenous bases. Those 4 bases are adenine, thymine, guanine, and cytosine, usually abbreviated A, T, G, and C. This is the 4-letter alphabet the entire genome is written in. The sugar and phosphate of each nucleotide link together to form a long chain, called the sugar-phosphate backbone, and the bases stick out from this backbone like rungs waiting to connect to a partner. Crucially, these bases pair up in a very specific way. A always pairs with T, and G always pairs with C. This is called complementary base pairing, and it's arguably the single most important rule in all of molecular biology. Everything from replication to reading genetic code depends on it. Put those base-paired nucleotides together in a long chain, and you get 2 strands running alongside each other, connected by their paired bases, like a ladder. Now twist that ladder around its own axis, and you get the famous double helix shape of DNA. One more important detail: each strand has a direction. Chemists label the 2 ends of a DNA strand 5' and 3', based on the structure of the sugar at each end. The 2 strands in the double helix run in opposite directions. One strand goes 5' to 3' left to right, while its partner runs 3' to 5'. We call this anti-parallel. This directionality matters enormously in practice. It determines which direction enzymes read and copy DNA, and you'll see this concept resurface constantly once we start writing code, because we always need to know which direction a sequence is being read. Zooming back out, how is all this DNA actually organized inside the nucleus? The full set of DNA in an organism is called its genome. That genome is divided into separate, tightly wound packages called chromosomes. Humans have 46 of them, organized in 23 pairs. Within each chromosome, specific stretches of DNA are called genes. A gene is essentially a set of instructions for building one particular protein, or sometimes an RNA molecule with its own function. Humans have roughly 20,000 genes spread across our 46 chromosomes. Think of the genome as an entire encyclopedia set. Each chromosome is one volume of that encyclopedia, and each gene as one specific entry or article within a volume, containing the instructions for one particular product. Not every part of a gene actually codes for protein, and not every part of the genome is a gene at all. Within a gene, the protein coding segments are called exons, while the segments in between, which get removed before the final protein instructions are used, are called introns. Only exons ultimately carry through to build the protein. Introns are more like rough draft material that gets edited out. Zooming out further, only about 1 to 2% of the entire human genome actually codes for protein at all. The rest is non-coding DNA, which for a long time was dismissively called junk DNA, but we now know much of it plays important roles, including regulatory elements, DNA sequences that don't code for protein themselves, but control when and how much a nearby gene gets used, acting like dimmer switches and on switches for genes. Every time a cell divides, it needs to make an exact copy of its entire genome. This process is called DNA replication. It relies on the same base pairing rule we already learned. A pairs with T, G pairs with C. To replicate, the double helix unwinds and separates into its two individual strands. Each original strand then serves as a template, a pattern, for building a brand new complementary partner strand. Because each new double helix ends up with one original strand and one newly built strand, we call this semi-conservative replication. Half of the original is conserved in each copy. The enzyme responsible for actually building the new strand, by reading the template and adding matching nucleotides one at a time, is called DNA polymerase. This elegant mechanism is why DNA replication is remarkably accurate. The old strand acts as a reliable blueprint for the new one. Here's a subtlety that trips a lot of students up. Remember, the two strands of DNA are antiparallel, running in opposite directions. But DNA polymerase can only build a new strand in one direction, from the 5' end toward the 3' end. So at the Y-shaped point where the helix is unwinding, called the replication fork, one new strand can be built smoothly and continuously in one long piece. We call this the leading strand. But the other new strand has to be built backward, away from the fork, so it's synthesized in short, disconnected chunks called Okazaki fragments, which get stitched together afterward. This is called the lagging strand. A helpful animation here would show the fork opening up like a zipper, with one strand drawn as one continuous line, and the other as a series of short segments being added and then joined. This asymmetry is a direct consequence of DNA's directionality that we discussed earlier. Even though replication is remarkably accurate, it isn't perfect. Errors do occasionally happen, and these are called mutations, permanent changes to a DNA sequence. There are 3 basic types. A point mutation is the simplest. A single base gets swapped for a different one, like changing one letter in a word. An insertion adds one or more extra bases into the sequence that weren't there before. A deletion removes one or more bases from the sequence. Point mutations tend to have a limited localized effect, since only one letter changes. But insertions and deletions can be far more disruptive if they occur within a gene, because they can shift what's called the reading frame, the grouping used to interpret the genetic code, throwing off everything downstream of the mutation, a phenomenon called a frame shift. We'll explore reading frames properly in our next session, but it's worth previewing here because it explains why insertions and deletions are often more damaging than simple point mutations. These aren't abstract concepts. Mutations have direct, well-documented consequences for human health. Sickle cell anemia is caused by a single point mutation in the gene for hemoglobin, the protein that carries oxygen in red blood cells. That one letter change alters the protein shape just enough to distort red blood cells into a sickle shape, causing pain and circulation problems. Cystic fibrosis is most commonly caused by a small deletion in a different gene, disrupting a protein responsible for moving salt and water across cell membranes, leading to thick mucus buildup in the lungs. And Huntington's disease is caused by a different kind of mutation altogether, a repeated sequence of DNA that expands beyond its normal length over generations. These examples make a critical point stick. A change as small as a single base out of 3 billion can have a profound impact on health, which is exactly why sequencing and comparing DNA carefully is such powerful medicine. Let's move to the practical part of today's session and finally write some Python. In Python, we represent a DNA sequence simply as a string of texts, a series of characters, in this case restricted to the letters A, T, G, and C. On the screen, you can see a line of code, SEQ equals, then the DNA sequence in quotes. That's it. Behind those quotes, Python treats it just like any other piece of text. Once we have a sequence stored this way, we can ask simple questions about it. Typing len, open parenthesis, SEQ, close parenthesis, tells us how many bases long the sequence is. And because a string is really just an ordered list of characters, we can access any individual base by its position, or index, using square brackets. Seq bracket 0 gives us the very first base, since Python starts counting positions at 0 rather than 1. These basic operations, length and indexing, are the building blocks for everything we'll do with sequences going forward. Here's our first real bioinformatics function. Computing the reverse complement of a DNA sequence, meaning the sequence of the opposite strand, read in the correct 5' to 3' direction. This is something we do constantly in real analysis. Let's walk through the code. First, we define a dictionary called complement, mapping each base to its partner, A to T, T to A, G to C, C to G. Exactly the base pairing rule we learned earlier. Next, we build the complement of our sequence using what's called a list comprehension. For each base in our sequence, we look up its partner in the dictionary and join all those partners back into a single string. At this point, we have the complementary strand, but it's still in the same left to right order as the original. We need to reverse it to get the correct direction. Python makes this easy. SEQ bracket : -1 reverses any string. Put together, this function takes any DNA sequence and returns exactly what you'd find on the opposite strand. Let's slow down and go through that function line by line, because understanding exactly what each part does is more valuable than memorizing the code itself. Line 1 creates our lookup dictionary. Think of it as a small translation table sitting in memory. Line 3, the def keyword, starts a function definition. We're naming our function reverse complement and telling Python it will accept one input, which we're calling seq.line 4 is the busiest line. It loops over every single character in SEQ one at a time. For each one, it looks up that base's partner in our complement dictionary, and the join method glues all of those partner bases back together into one continuous string stored in a variable called comp. At this point, comp holds the complementary bases, but still in the original left to right order. Line 5 fixes that. The notation bracket : ::1 is Python's shorthand for reverse this entire sequence, and return sends that final, reversed, complemented string back out of the function. Try running this yourself on a short sequence and tracing through each step by hand. It will make the logic click. One more useful calculation: GC content, meaning the percentage of a DNA sequence made up of G and C bases. This is a genuinely important biological measurement. Regions high in G and C bind more tightly together than regions high in A and T, since GC pairs form 3 chemical bonds, while AT pairs form only 2. Higher GC content is linked to more stable DNA, and GC content varies meaningfully between species and even between different regions of the same genome, making it a useful signal in identifying genes or comparing organisms. Here's the code. We count how many times G appears in the sequence, plus how many times SCI appears, giving us the total G plus C count. We divide that by the total sequence length using len of SEQ and multiply by 100 to express it as a percentage. This single line calculation is something you'll reuse constantly. It's often one of the very first things a bioinformatician checks about any new sequence. Let's bring today's session together. We started at the level of the cell, distinguishing prokaryotic from eukaryotic cells and locating the nucleus as DNA's home. We then zoomed into DNA's chemistry, nucleotides, base pairing, the double helix, and strand directionality. We zoomed back out to see how the genome is organized into chromosomes and genes, and how genes themselves contain both coding exons and non-coding introns. We covered DNA replication, including why one new strand builds continuously while the other builds in fragments. We discussed mutations, point mutations, insertions, and deletions, and saw how single letter changes can cause real diseases like sickle cell anemia. And finally, we wrote our first Python code, representing sequences as strings and computing both reverse complement and GC content. Next session, we take the next logical step, how these DNA instructions actually get turned into working proteins. 3 ideas to hold on to from today. First, complementary base pairing, A with T, G with C, is the single rule underlying nearly everything we discussed, from the double helix's shape to how replication works. Second, DNA's organization is hierarchical: genome, then chromosomes, then genes, then exons and introns within genes, and keeping that hierarchy straight in your head will make every future topic easier to place. Third, even a single letter change in 3 billion bases can cause disease, which is a powerful illustration of just how much precision biology depends on, and exactly why the computational tools we're building in this course matter so much. Hold on to these 3 ideas as we move into gene expression and protein synthesis next. Time for a short quiz. 5 questions. 1. What organelle stores most of a eukaryotic cell's DNA? 2. Which base pairs with guanine? 3. What is the difference between an exon and an intron? 4. Why does the lagging strand get built in short fragments rather than one continuous piece? 5. Name one disease caused by a DNA mutation, and what kind of mutation causes it? Pause and answer each one in your own words before moving on. If any of these feel shaky, it's worth rewatching that section of today's session before we build on these ideas next time. For homework this week, first, write your own reverse complement function from scratch, without looking at today's slides. Then test it on a sequence of your choosing. Second, calculate the GC content for 3 different DNA sequences. You can find real ones on NCBI from our previous session, or use short made-up examples. Try to predict which one will have the highest GC content before you run the code, then check your intuition. Next session, we move from DNA into gene expression and protein synthesis, the process by which the instructions stored in DNA actually get read out and used to build a functioning protein. We'll cover transcription, translation, the genetic code, and protein structure, and in Python, you'll write your first program that translates a DNA sequence directly into a protein sequence. See you then.